All right, let's talk about the final section of Chapter 11, Section 4, which covers mostly the process of meiosis. Meiosis is the process that is used, that plays an important role in the production of these reproductive cells or gametes. Um, all right, but first, let's think about chromosomes. And so, as we know in people, we have in humans, we have 46 chromosomes, and they come in pairs, as you can see in this picture here. You have a pair of number ones, twos, threes, etc. And they're usually arranged in a picture like this from the largest to the smallest, with the sex chromosomes put last. And so this is the picture of the chromosomes from a male because this person has an X and Y sex chromosome. Of course, if they were female, they would be X and X. Um, So again, they're arranged in pairs, and these pairs are known as homologous pairs. So here's the homologous pair of number one chromosomes, homologous pair of number two chromosomes, etc. What do we mean by homologous? Well, as the prefix suggests, one or one and the same. So let's look at the pair of number two chromosomes here. What it means is that because they're homologous, those pairs have the same genes on them. Okay? They have the same genes. To be homologous means you're a pair of chromosomes that has the same genes. Now, you may have, as we've seen, the same form of the gene, big R, or you may have different forms of the gene, big G, or big T, little t. But a homologous pair of chromosomes all have the same genes on them. Okay, keep that in mind. Um, this condition of having homologous chromosomes, having two copies of each chromosome, it's what's known as being diploid. People, humans, we are diploid organisms. We have two copies of each chromosome, okay? Now, some organisms, like bacteria, are haploid. They only have a single chromosome, so they only have one copy of each gene. They are haploid. Now, we're going to see that in meiosis, we're going to use, or in the production of gametes, we're going to use meiosis to produce haploid cells. That is, our reproductive cells. In the case of male sperm cells, in the case of female eggs. These things are haploid cells, and we'll see how that happens and why it happens. There's a way of abbreviating things that are haploid and diploid. If a cell is said to be 2N, that's the diploid condition. N is the haploid condition. Okay, so twice N would be diploid. That's just an abbreviation for haploid and diploid. All right, so let's think about meiosis. Meiosis is a process that just happens in the organs in your body that are responsible for producing um, the reproductive cells. In the case of females, the ovaries, there are cells that go through meiosis, and in males, the testes, there are cells that go through meiosis. Doesn't happen in other parts of your body, just in your reproductive organs. So let's look at meiosis. So like all cells, the cells in your reproductive organs can be found in interphase, and most of them will be in interphase, just hanging out. But when those cells have been told to divide and to make gametes, these haploid reproductive cells, they, instead of going through mitosis, they go through meiosis. And so let's look at what's going on here. In meiosis, we have two divisions. Unlike mitosis, where there's just one division of the cell, in meiosis there's two. You go from one cell to two, and then two to four for a complete cycle of meiosis. So, meiosis 1, we have prophase 1, metaphase 1, anaphase 1, telophase 1. Let's look at what's going on in prophase 1. Well, like mitosis, the nucleus breaks down, okay, the nuclear envelope. At this stage, the cell has gone through interphase, so we have duplicated chromosomes. Notice each chromosome consists of two chromatids, okay? In our example here, we just have 4 chromosomes. To keep it simple, we've got 2 big ones and 2 little ones. The 2 big ones are homologous, and the 2 little ones are homologous. Now, in prophase 1, that is in meiosis, something unique happens here. And what happens is the homologous chromosomes get together and they form what's called a tetrad, meaning you basically have 2 chromosomes with 4 chromatids kind of gathered or bunched up. And so at this tetrad stage, what happens is this process called crossing over. And you can see what happens is the homologous chromosomes will exchange pieces of DNA. And so notice that, let's say in this individual, this was the chromosome they got from their dad, and this is the one they got from their mom. Notice that after crossing over, you have a chromatid that has some DNA that originally came from dad and some that came from mom, and here's one where you have some that came from mom and some that came from dad. This is sort of the beauty of crossing over during prophase 1, is it takes DNA that came from mom and dad and mixes it around, essentially creating genetic variation. Very important process here. In metaphase 1, these homologous pairs of chromosomes, these tetrads, line up. Recall that if this had been mitosis, the duplicated chromosomes all would have lined up and they would have been pulled up. But in metaphase, we have the homologous pairs lined up, okay? Not the individual chromosomes, but the homologous pairs. And notice during anaphase, you now have the homologous pairs pulled apart, okay? Not the chromatids, but there's one chromosome of homologous pair, here's the other one, and they are pulled apart, not the chromatids. So, by the end of meiosis 1, when we get to telophase and we're reforming the nucleus, we still have duplicated chromosomes present, but notice again, some of them are mixtures of DNA from mom and dad. And notice what's happened. We started out with 1, 2, 3, 4 chromosomes. At the end of meiosis 1, we only have 1, 2, 1, 2, 2 chromosomes in each cell. Essentially, the cells after meiosis 1 have become haploid, or N. We've reduced the number of chromosomes at the end of meiosis 1. Now, after meiosis 1, we've got 2 cells now, and they are haploid cells. Each of their chromosomes is still in that duplicated state consisting of 2 chromatids. Now, meiosis 2 is a lot like mitosis. We're going to take these 2 cells and divide each of them. And so in prophase, again, the nucleus breaks down, the centrioles split in 2 and go to opposite ends. The spindle fibers attach to the duplicated chromosomes. During metaphase, we line up those duplicated chromosomes. During anaphase, we pull apart the chromatids. And so then, by the time we get to telophase and then cytokinesis, we now have 4 cells, each with their own nucleus, of course, and each with the haploid number of chromosomes, but we don't have duplicated chromosomes at this point because the chromatids have pulled apart. So let's compare mitosis and meiosis here. So again, in mitosis and meiosis, we've gone through interphase, and in both situations we have these duplicated chromosomes. In prophase, the chromosomes condense, but a big difference here is in meiosis, those homologous chromosomes get together and do crossing over. That does not happen in mitosis. Metaphase of mitosis, all the duplicated chromosomes line up. In meiosis, the homologous pairs line up along the center. Anaphase of mitosis, the chromatids are pulled apart, and over in meiosis, It's the homologous pairs that are pulled apart, not the chromatids, okay? So at the end of mitosis, we have two identical cells that are diploid. At the end of meiosis 1, we have two haploid cells that are different than each other because of this phenomenon of crossing over, okay? They are different from each other, genetically speaking. So then we go through meiosis 2, which, like I said, is a lot like mitosis, where we take these two cells we've made and split them into four, and they are each haploid cells. So yes, we have haploid cells, four haploid cells we produce by the end of meiosis. They're all different from each other, genetic variation, and so in reproduction, basically, one of these gametes from mom will get together with one of these gametes from dad. In the case of people, mom's egg containing 23 chromosomes, dad's gamete containing 23, and when they get together through the process of fertilization, you make a zygote that has 46 chromosomes. So this is why in meiosis we need to produce these haploid cells, because two of them are going to get together to form the next generation, and so you want to have the same number of chromosomes from one generation to the next. All right, last in this section, I just want to briefly talk about this phenomenon of genetic linkage. And so we saw with all the Mendelian stuff and the traits he looked at on his pea plants, they all adhere to this idea of independent assortment. And again, that was because those traits are found on different chromosomes. But as I've mentioned in class, in us, we have over 20,000 genes, but we only have these 23 pairs of homologous chromosomes. So you've got to have on each chromosome a lot of genes. And so, for example, here we see in a fruit fly, this chromosome number 2, here are just a few of the genes that are on this chromosome. So when you have genes that are on the same chromosome, this is known as genetic linkage, or those genes are linked. And when you have genes that are linked, essentially independent assortment doesn't apply. They don't sort independently. They are linked to each other. Okay. All right. So that's it for section 4 and chapter 11.